we're planning on reporting. All right, so I'm just going to do a super quick introduction. So thank you everybody for coming in for a special seminar time, and to Dr. Mishra for rearranging his schedule in the semester because of unpredictable and tragic events. But Dr. Mishra is here. He's a part of the Department of Statistics at UGA, and he was at MD Anderson, and he's going to give us an introduction to his work and how that relates to IOB. So thank you very much.
Okay. Thank you for the introduction. Thank you for, first of all, the opportunity. I recently joined UGA around less than a year back, so I'm still trying to learn the underlying research around multiomics and things like that. And in that, so with that quest in mind, today I'm going to talk about my research work, more focused on leveraging multivariate analysis approach for modeling high-dimensional multiomics data sets that I believe that some of you might be encountering these days. Okay, so to begin with, as far as this multimodal data sets is concerned, or when we talk about multimodal data sets in biomedical research, we often talk about data sets collected in a given trial or a study from different sources. And it can come from anything like, it can be omics data sets, metabolites data sets, microbiome, EHR data sets. Like when a patient basically go to any of these hospitals, they share lots of information about their past medical history and so on like that. And so EHR data now gets collected at petabytes level. And then we have wearable data and all sorts of things. Environmental data is again, like now in the context of climate change, when we are talking about things like it is coming out to be very, very relevant, like as far as the modeling or statistical modeling aspects is concerned. Then the idea behind when we have all these data sets collected from different sources, when we talk about multimodal data sets, the idea is that how to integrate information from all these data sets and efficiently use it for either for precision health or like, biomarker discovery and things like that, that you might be doing on a daily basis. See, even at the cellular level, right? So when we talk about things like disease, like cancer and so on like that, we often talk about, okay, cancer is like malfunctioning of the cell. So we want to understand everything about like what is happening when a cell goes bad. And in that respect, we often talk about like collecting data sets like proteomics, lipidomics, metabolites, microbiome, single cell RNA-seq data sets, all of these things. When we talk about what are these like data sets collected from different sources actually tries to give us. So I think this cartoon gives us like a great introduction about when we talk about all these omics data sets. In principle, you can think of like there is a giant elephant and there are lots of people, like everyone is kind of blind and he or she is basically trying to touch a portion of the elephant and then trying to guess like, okay, what might be happening or who this particular object or giant beast is. And everyone kind of tries to get its own guess, like, okay, someone will say that, hey, it's like something like a seer or like tree and things like that, just by looking at one aspect of the data sets. Something in similar flavor, like all in one sense, if you think about it, what each of these persons are seeing is basically an orthogonal or some or partial overlapping view of the same system. That is this particular object, and something that we can relate it to when it comes to omics data sets or multimodal data sets in case of medical research. All of the different omics components or the EHR data or the medical records data, they try to give us some information. In some sense, they try to give us like some overlapping information, but at the same time, there is a lot of it like which is highly orthogonal, like not at all dependent on each of these things. But to get a good inference out of all these data sets, one good approach would be is that integrate information from all of these sources and then make a guess like what this particular species is. And then in that context, the idea of statistical method development comes in, and that I'll introduce later. But with that particular idea in mind, now you see many study appearing in leading journals, which tries to collect all of these data sets from different sources. And the aim is that how to integrate information from all these sources to get some good idea about the underlying biological system. For instance, this paper came in immunotherapy melanoma, where they wanted to study the role of probiotics and fiber in cancer patients, and appeared in Science. So if you see like the kind of data collection effort that they did in this case, it ranged from all sorts of omics component. The idea is to get all the different kind of view of the underlying system and integrate it. And it need not to be cancer, like things like colorectal cancer study appeared in Nature Medicine, right? Again, the data sets that got collected in this particular set, ranging from metabolites to proteomics to microbiome data sets and so on like that. As a statistician, we believe that, oh, this is like really rich data sets, and something needs to be done to basically extract information from all these data sets. This is again like in case of, as a part of this integrated human microbiome project, and they are trying to basically, as a part of this particular project, they are targeting different disease, and IBD or inflammatory bowel disease is just one disease. Again, the idea is to, in a crowdsource, collecting data sets from different individuals who are affected by this particular disease in a crowdsourcing way, and then again collect data sets as far as different omics data is concerned. Okay, given that all of these, statistically speaking, like whenever we see a biological question, we try to understand like, okay, what is it the data or the scientist is trying to tell, or what kind of hypothesis that that scientist has, and how us as a statistician will basically bring in those hypotheses and develop, either use existing tool efficiently or develop methodology around it to extract information based on the hypothesis that scientists basically guides us. Now, in the larger framework of integrative statistical learning, the idea is to integrate all of these orthogonal views collected from different sources and come up with a statistical approach that allows us to churn all these data sets together and then like come up with interesting biomarker, interesting understanding about underlying system and so on like that. And some of these things you already might be doing in your research work. Today, what I'm going to talk about will be more focused on around like multivariate, how multivariate analysis approach can help us to do this integrative statistical learning in the case of multi-omics data sets. And some of you like might be already familiar with what is the basic multivariate analysis framework will be. In a given multivariate analysis framework, for instance, if you are like talking about multivariate regression framework, it simply talks about understanding the association between multivariate outcome Y and X. And then in terms of the coefficient matrix C star, it tries to understand the strength of association. And accordingly, like we do inference on the strength of association, and that basically gives us the idea about what is happening in the underlying system. Multivariate analysis approach not only just restricted to multivariate regression, things like dimension reduction techniques, like or multidimensional scaling technique that you do on a day-to-day basis, basically encapsulate the larger framework of multivariate analysis. Now, there are already existing framework, and now when we see all of these omics data sets coming from different sources, the classical multivariate analysis framework, when it was developed, was thought about the methodology in a very, very ideal setting. But when it comes to multi-omics data sets, we see all sorts of anomaly related to the omics data sets, ranging from sparsity, dynamic association, missingness is there, then heterogeneity among the populations or the samples that you have collected, and high dimensionality is the giant piece, right? So often if you collect microbiome or metabolites data, in principle, you are observing thousands and thousands of metabolites. So high dimensionality is a big problem. And contamination and nonlinearity and all of these things are there. And in my research so far, in the larger framework of multivariate analysis or multivariate framework, I've tried to come up with different statistical approach that in principle basically allows us to integrate information from all of these omics component with certain, obviously not solving the entire problem, but partially solving one problem at a time using the framework. using the framework. So for instance, my journey started with the sequential post-sparse factor regression model, or like generalized post-sparse factor regression model, with the idea of solving the problem of high dimensionality and the missingness in the response. So I'm not saying that I'm solving all the problem of multi-omics, but I'm just saying that, like, okay, there are certain aspects or complexity of the omics data sets that in each of these models basically allows us to address those issues. So one approach tries to help us with the high dimensionality and missingness. Then we kept on developing where we said, like, okay, now we have high dimensionality missingness. We can also let us try to develop or build upon the methodology where it can handle the mixed data types and so on like that. In a moment, we'll see like what kind of applications that basically motivated from. Then count data sets and over dispersion, like with anyone, like if you work with the transcriptomics data sets, we often come across with the genetic expression data sets more at the, or if you, with bulk RNA sequencing, what we have is basically the count of the abundance of these different genes. And then like there is obviously an over dispersion. There are some standard tools that is available, but then like when we try to think of analyzing all of these data sets jointly, that's where the problem comes. And so in the approach, like we try to address one aspects of the complexity of the data sets over the period of time. With factor instrumental variable analysis approach, like we are trying to now answer the questions around the causality, like what kind of causal set of microbiome or metabolites or like genes that are basically associated with the phenotypes of interest and so on. Yeah. Okay. So to begin with, right, so starting with the, just to give you some flavor of like the motivations, the biological motivations behind the methodological development. So for instance, in the genomics application, one common question we often ask ourselves, like how the transcription factors basically regulates the genes expression data sets. So initially the journey started with this data sets coming from yeast cell cycle study, where the idea was that like for each of the in case of yeast cell experiments, we observed thousands of genes at multiple time points, and corresponding to that, we also had data about the transcription factors and its expression value. And the idea is that like how transcription factors regulates genetic expression at multiple time points and so on like that. So the data looks like something like really this, right? So on one side, like you have all these genes, their RNA levels collected at multiple time points. And then like on the other side, you have a lot of transcription factors and their expression levels and so on like that. If you think about it, like, okay, if I want to understand the association between transcription factors and the RNA expressions at multiple time points, it seems to be like a regular multivariate analysis problem, like, so where you can think of like, okay, let us use a classical multivariate analysis framework and do an association and use the coefficient matrix to basically understand the association between transcription factors and genetic expression data in a multivariate sense. But the challenge that comes from the high dimensionality, right? So when I say high dimensionality of transcription factors, so in principle, we all know that talking to scientists, like we always say that many of the transcription factors tends to act together to affect genetic expression data sets. It is not that at any given time point, all the transcription factors are active and basically regulating the gene expression data. So the idea of when we say the high dimensionality of transcription factor, we are talking about a set of problems in statistics, which is called variable selection, where we say that not all transcription factors are important. Maybe only a subset of those are transcription factors are important, and it is regulating the genetic expression data sets. And same thing about the multicollinearity, that saying that it is not just one transcription factor that will affect the genetic expression data set. Rather, these transcription factors are acting in tandem. We don't know what combinations are, but the biological motivation, it says that it is acting in tandem to basically affect the genetic expression data set. So, and many of the transcription factors are obviously working in coherence, leading to the problem of multicollinearity as far as the statistical notion is concerned. As far as the outcome side, the RNA levels is concerned, there are the RNA levels at multiple time points. They are again, like, since we are observing those RNA levels in the same genes, at same genes as multiple time points, because they coming from the same genes, they are often related and so on like that. So, and a classical multivariate analysis framework are not equipped to handle these kind of statistical challenge, like for instance, variable selection, multicollinearity, or dependent outcomes. That's where we propose the idea of more when talking about translating the particular challenging idea in actual scenario, and suppose that we are looking for a set of transcription factor that is acting in tandem to affect a set of genes or set of gene or genes at a set of time points and so on like that. Statistically speaking, I can translate this particular idea in learning. So you have two things, like Y is, think of it like your RNA levels, and then X is like transcription factor. When I say that only a set of transcription factor is acting together to affect a set of genes, ideally, I'm interested in learning some U1, which is kind of indicative of selection of transcription factors, and some V1 is again like indicative of selection of time points in where a set of transcription factor is affecting genes expression at only these two set of time points and so on like that. This is mathematically, this looks like if I am able to somehow guide my analysis looking for U1 and V1, I'm done. Like this is what my scientist has told me to look for, and I can get this thing using this kind of formulation. But it seems that like, no, there can be multiple such type of association. So U1 and V1 can identify one such underlying association, but at the same time, you have V2 and U2 that can look for another orthogonal association that might exist in the data sets. When we put all of these things together, you are essentially looking for, you have one outcome and variable outcome and predictors, and you're looking for U1 and V1, U2, V2. But essentially, what U1 and V1, what we want to emphasize over here is that U1 and V1 needs to be sparse, then only we can make a good interpretations. A sparsity is very, very important for us to select on the transcription factors or like the genes expression at a subset of time points and so like that. When you put all of these things now together, you are essentially looking for understanding Y in terms of X, and if you have U1, U2, and V1, V2, your job is done. And if U1 and V2, U1, U2, V1, V2 is sparse, you're not only able to express the underlying association in few signatures like U1 and V1, but also identified in terms of the sparsity. Statistically speaking, this is called learning in a classical multivariate regression framework, where we talked about coefficient matrix, where there was no restriction at all on the coefficient matrix. Now, in this case, we are putting, because of the underlying biological phenomena, we are putting a restriction on the coefficient matrix. Now you are saying that you cannot be free at all. You want to be, now this coefficient matrix should be a combination, should be expressed as a combination of U1, V1 plus U2, V2. And essentially speaking, or statistically speaking, this is called low rank and sparse coefficient matrix. So initially it was guided through the underlying biology, but at the end of the day, we want to frame it like a statistical problem, a valid statistical problem, and look for like what kind of coefficient matrix or what kind of regularization that I want to make. Okay, so given that coefficient matrix is low rank and sparse, and it is desirable also, in one sense, to understand what this low rank and sparse coefficient matrix, why, first of all, it is desirable, but what essentially it is trying to do. So when I say that the coefficient matrix, it is of P cross Q dimension, if I let it to be free, then it will be of really, really high rank. Imagine that your original image is like a coefficient matrix. So any image can be expressed like a two-dimensional matrix with some values in it. A same image can be represented in some low rank approximation. So for instance, if you approximate that particular image with rank 2, it is very, very blurred. But if you go on increasing the rank and so on like that, you are able to say that like, okay, with sufficient or with only 14 rank or 14 set of linear combinations, I can basically regenerate that particular figure, which is good enough for doing any analysis. In statistics or in image processing, this is called like the compression of, like the image compression. I'm trying to express this giant information in the original image in few latent factors and so on like that. So essentially, when we say in the multivariate analysis framework, I'm trying to look for a coefficient matrix which is low rank and sparse. I'm not compromising on the information, but rather I'm regularizing them to learn from each other and so on like that, motivated from the biological questions and so on. And obviously, with sparsity, we get the interpretation aspects. Okay, so once we have low rank and sparsity constraint, I'll go fast on the mathematical part. So expressing the low rank and sparse coefficient matrix is equivalent to saying that regression y in terms of x, where it is decomposed now as u times d times v transpose, where u is essentially sparse. Like, so 0 is essentially saying that like extract me the R number of latent component or latent combinations where each of the columns of U can be sparse. And similarly, extract me V, which is in principle, again, like it should be sparse in one sense, because the sparsity is very, very important for us to make the underlying inference. It seems that there is a computational challenge associated with this particular kind of, so from in a classical regression framework, though it looks good, but the challenge is more arises from estimating this particular thing. Proposing an idea is always good, but when it comes to like dirtying your hand, like when it comes to estimation and all, that's where the challenge arises. So in this particular paper, we basically overcome the issue of challenge of estimating these low rank and sparse coefficient matrix uniquely. And we proposed co-sparse factor regression model with a formulation. And then we proposed basically a computational approach called sequential co-sparse factor regression model to estimate the required low rank and sparse coefficient matrix. That's when then we can use it for the inference and so on like that. So I'll skip through this particular thing quite fast. But let us, when we applied this particular method on the data sets, again on the each cell genetic expression data sets, we kind of identified multiple. So the model basically identified three latent. The rank of the coefficient matrix was identified to be 3. And then each rank, or corresponding to each rank, we identified some set of transcription factors, which is affecting a genetic expression at a given set of time points. So out of thousands of transcription factors, each unit rank basically identifies. So for instance, first component basically identified 61 transcription factors, second 38. So out of thousands, you are now reducing your analysis to only focused on few of the transcription factors. And compared to the existing statistical approach that in principle this could have been possible, we were able to identify, obviously in terms of the prediction accuracy and all, like we performed better, but also qualitatively, like we identified more transcription factor that in principle affect the genetic expression. So for instance, like as far as the different transcription factors is concerned, when we try to look for how it loads or how it is basically moving in time, and we kind of find out a nice cyclic pattern around its role of a given transcription factor, giving us some idea about the underlying cellular process in genes, like maybe something is happening, either cell division or something like that at regular time point, and that allows us, that again gives us a nice perspective to scientists to look for what is happening at these 2 time points, and maybe we want to understand those things. Like the loadings, like something how you interpret the principal component loading matrix. Again, it is giving you each of these transcription factor is, you can think of like a bag of transcription factor and acting together it is affecting the genetic expression data set. So the methodology allows you to look for the role of individual transcription factor, how it works at multiple time points, but at the same time, through a combination, through each of, we can interpret each of the transcription factor as a bag of transcription factor and how it is working together in tandem to affect the genetic expression data set. Coming on, right, so this project was really important for us to understand the strength of low rank and sparse coefficient matrix, and then leveraging on top of this on top of this particular structure, as it allows us to interpret a lot of things from big data sets to in a very, very small framework, in a smaller setting or in a reduced setting. We try to extend this particular approach in other settings as well. So for instance, this particular, the second application mode came from the public health application mode coming from the longitudinal study of aging data sets, where the idea was that like we had data sets so the aging population were observed at mainly 2 time points. And we were interested in that how is it that the past medical condition or past economic status is affecting the future medical conditions and so on like that. And when we processed this particular data sets, like what we had was basically more than 3,000 samples with 294 predictors, and these predictors were the important thing about this particular data sets, like we were interested, we considered the future medical condition, which were mainly 44 variables as my outcome variable, and some predictors related to the past medical condition and economic status as a predictor variable. But if you see this particular data sets, the challenges were more coming from when we said like we had data from future medical condition, and those were related to the health status, memory status, depression status, primary activity, cognitive ability, or sensation condition. Out of this 44, 3 outcomes were of continuous type, and 44 of them, or 41 of them were of binary types. The power of doing multivariate analysis is that, and with the regularized structure, is that it kind of forces them to learn from each other. When you analyze each of these outcomes individually or independently, you are not forcing them to learn from each other. Because of the kind of regularization structure that we put on the coefficient matrix, it is forcing them to learn from each other, and it increases the power of the model or power of the model to extract information that might exist in the data sets. So, and given that with this particular notion that no, we want to make them learn from each other as well as want to do a good interpretations out of it. So we said like, no, we want to analyze both continuous and the binary data sets together in the multivariate analysis framework. And that's where this generalized co-sparse factor regression model goes. And so in addition to high dimensionality and the missingness in response, what this model was able to do or can do is basically handle mixed outcome type, and something in the case of multi-omics or multimodal data sets, when we talk about integrating EHR data sets and so on like that, we often see outcomes are of multiple types and so on. And if we want to analyze them together, these kind of models in the larger multi-omics framework can be like really, really helpful, where you don't need to throw away any of the categorical or the continuous outcome or analyze them individually or marginally. Rather, you can throw everything in this particular giant model and let the model do its job itself. It will do selection of important predictors, important outcomes for you under the hood itself. If there is signal in the data sets, then it will extract. And also, like one caveat of all these models is that it needs to have sufficient sample size to extract information in the data sets. That's like a given. Okay, so again, like we can look at the usefulness of a particular approach in the larger context of multimodal research, where we can now think of like integrating omics data sets with mixed type of outcome. It can come from wearable or like EHR data sets is a great source of obtaining mixed type of outcomes and so on like that. And if you want to integrate that in a given multivariate framework, this can be like really, really helpful.
Aditya, have you done any sort of estimation of what the minimal sample size is?
Sorry?
Types of analysis. Minimal sample size that you can have.
For like it requires sample size, so for instance, it varies. Like when we do microbiome metabolites association, even 100 sample size is coming out to be like where the strong signal is there. So even it can happen like even if you have 100 sample size, but if there is a strong talking going on between two multi-omics component, even 100 sample is sufficient enough. But if there is so when we do the simulation analysis, we try to show that in terms of more signal to noise ratio, that how much signal that your true model have and how much noise that you have, and how when we increase the different model noise, how the estimation deteriorates and so on like that. So sample size, even a small sample, to answer your question, like even a small sample size, if the signal is really strong, which we generally see in the microbiome metabolites case, it kinds of learn or extract the component or underlying association. Going on, right? So my third application is around the microbiome data sets, and this is where I work a lot around. My journey of multiomics started from microbiome. First it was genomics, but then for a long period of time it is microbiome. To give you some flavor about the microbiome data sets, like microbiome is all over the place. It is in soil, in ocean, in gut, and now as we are learning more and more about microbiome, we want to understand its integral role in different contexts, be it human and so on like that. And my work at MD Anderson were more focused on understanding microbiome in gut, but my past work was more focused on microbiome in the ocean. So a way to collect these microbiome data sets, you collect sample, you do RNA, you can do metagenomic sequencing or PCR, 16S RNA sequencing to identify in a given sample who all of these species are. And once you process this particular sample using any bioinformatics pipeline, the idea is to collect their abundance data sets. And then the challenge is again, like as far as this microbiome data sets is concerned, a huge, all the regular task of statistical tasks ranging from classification, regression, it needs to be done. We try to formulate those kind of statistical problems. But the challenge mainly arises from, if you have seen microbiome data sets, it is really, really high dimensional, very, very sparse. You will see some species appearing as the technology evolves. We are now going more and more granular at the species level. Earlier, it was more at class and order level. But even if you see any regular microbiome data sets, there are a lot of problems around. Now, first of all, it is count data set, over dispersion, lot of zeros and so on like that. And the idea is, again, like the statistician basically have their bread and butter around the complexity of the data sets. Like if the data set is complex, then we believe that, like, okay, that's where the methodology needs to be done. Okay, so this particular application is more coming from American Gut Project. It is again a crowdsourcing project where until this particular date, there are more than 30,000 microbiome samples were collected, and these samples were collected from different parts of the body. So people basically donated their samples, ranging from fecal, oral, hand, skin, and so on like that. And then after that, what we had in this particular project is basically the microbial abundance data sets. For our purpose, and in addition to the microbial abundance data sets, though we have 30,000 samples, but the project also tries to collect their habits, like feeding habits, what kind of food that they eat, and so on like that, what kind of demographic behavior or demographic location where they come from, and all sorts of information about it. So for our analysis purpose, we mainly focused on a subset of samples where we believe that there are sufficient informations, both in terms of the microbiome as well as a set of predictors is available. So we basically pre-process the data. For our analysis purpose, we mainly focused on 637 samples, and we aggregated the data at the family level to see some definite signal in terms of the microbiome. And then, like what we had in terms of some of the features that we were mostly interested in, diet-related features, and those data that were collected using BioScreen, we believe that this can be like really, really great data sets, less chance of error, and so on like that. So we mainly focused on those set of data sets. Our simple question was, like, can we understand the association between features mainly related to health condition, diet, demography, nutrients, and habit, and its association with microbiome, in addition to obviously some of the demographic indicators ranging from age, BMI, and genders. One thing, so obviously with microbiome data sets, there are a lot of challenge. I'm not saying that I'm solving all the problem, but all I'm addressing in this particular data sets in the larger multivariate framework, I'm trying to address the challenge of count data sets, which is over dispersed. So we mainly focused on these two set of these two attributes of the microbiome data sets, and that is one of the main reason we aggregated data sets at the family level, because we are not solving the sparsity problem in this particular methodology. So with focus in this, but with focus on overcoming the challenge of over dispersion, count data sets in a multivariate framework where you want to leverage upon the basic idea of low rank and sparse coefficient matrix, want to learn some latent combinations of like features which is affecting a set of microbiomes. That's where our focus was, and then we developed this particular methodology. By the way, all the methodology that I have shown, like it is already, the paper is published, there is a computational R package associated with it, and in principle, I believe that like with minimum modifications with your processing, with your data sets and all, in principle, if the data set has those kind of constraints, the models or the package can be used for analyzing the data sets. Since I'm already here, like I would always love to hear if you have any such data sets, like where you think that like my package or methodology can be used for analyzing the data set. Okay, continue. And when we have this, when I say count data sets, so this model was well equipped for understanding or modeling the count outcome or joint modeling of count outcome in terms of the set of features. If you think about this larger multi-omics framework, when we talk about count outcomes, like single cell RNA-seq and the transcriptomics data sets often have the genetic abundance data set, which is of count outcome. Over dispersion is there. And then we often want to understand how the different transcription factors or transcript expression is associated with either can be epigenetic variables or like any other omics data set. It can be proteomics or lipidomics and so on like that. So we see great potential of these kind of methodology now in the larger framework of multi-omics. And obviously, the motivation for methodological development was one, but then we can take this thing further to many, many, many applications. And so this is, so this is another application that I want to talk about, like where we took the game further. And application was mostly motivated from, and this is the latest addition in this larger, bigger cog of wheel around multi-omics, where my application was mostly focused around hepatocellular carcinoma study. And as far as this particular study is concerned, like to know some facts about this particular HCC, they're like 75% of the liver cancer are basically focused around this. What happens, there are many risk factors associated with this particular kind of cancer, like hepatitis B conditions, like NAFLD, like non-alcoholic fatty liver disease, or non-alcoholic NAS, or state of hepatitis, or like if there are certain genetic disorder, there is, or exposure to aflatoxin, like these are some of the risk factors that basically increase your chances of getting liver cancer. The bad thing about this particular type of cancer is that it is not detectable until like really, really late. And so often, like when we are trying to understand this particular type of cancer, poor mouse is basically our model, and then we try to understand how, like, what are the different stage in which this particular type of cancer manifests into, first of all, in a given animal, and then like obviously the humans. So apparently the manifestation of HCC, it has multiple stages going from a normal liver to different stages of, so when there is a chronic inflammation for a longer period of time, it passes on to the multiple stages, and ultimately it manifests into what we see now, HCC or hepatocellular carcinoma as a cancer. So what this particular study, and given that the complex nature of this particular type of cancer, we were fortunate enough, or at MD Anderson, like we were fortunate enough, like to get this data from this Nature paper from Subnam Salapurs, like, and she's trained as also as a statistician. So the good thing about this particular study is that she collected more than 400 samples. So given that because of her training as a statistician, she believes that no, if she do not have sample size, she will not be able to extract the signal. So given that she was statistically aware, we find this particular study like really helpful when it comes to methodological development or digging this particular data further. Okay, so in this particular study, again, like in the case of mouse model, like they basically introduced mice with okay. So for around more than 408 mice, what they collected, and they did a lot more work. I'm just presenting one aspect of the data set that she collected. One was a set of microbiome data, around 710 taxa, and set of metabolites, more than 7,000 metabolites data sets. And as far as the different stages of or different phenotypes related to this particular HCC cancer is concerned, she was mostly interested in the IGA, obesity as an outcome, NAS, HCC is obviously the final outcome, and different those are like different indicators related to immunology, and some of them are T cell, B cell, and some of them, these are all indicator variable, like yes and no. And she was asking a simple question that, okay, how my microbiome and metabolites are basically affecting, or how my phenotypes are associated with these microbiomes and metabolites. Now, if you see the standard set of statistical approach that is available to do this particular job, like we often look at things that like, okay, be it like with the microbiome data sets, some of you might have seen use DSeq or LEPC analysis or ANCOM-DC analysis to understand or identify a set of microbiomes that is affecting or that is differentially abundant as far as a given phenotypes is concerned. And same thing for analyzing the metabolites data. There are techniques like the GLM-based technique or PLS, partial least square differential abundance, PLSTA or ANOVA analysis, which mainly focused on identifying a set of or a different set of or a set of metabolites that is affecting or that is affected by these phenotypes and so on like that. But there is a fundamental catch in here. Essentially, when she generated this particular data sets, her question were not that how phenotypes is affecting microbiome. Her question were mostly like how microbiome is affecting phenotypes. So the directionality is very, very important here. And how my different interventions, how my metabolites is basically affecting the phenotypes of interest. And those were all like, again, related to HCC status, type 1 diabetes, or like NAS and so on like that. So the directionality is something that we want to focus in here. So with this particular idea in mind, that what she intended to do and what are the statistical approach that is available that in principle people tend to use. So given that, taking her hypothesis in the right direction, what essentially I am interested in, again, the same question, microbiome and metabolites, now in opposite direction with a set of extra condition that often at time when we try to say that, okay, microbiome is affecting phenotypes of and so given that directionality is right, there are already methods available for it, but it is not that microbiome is acting in whole and soul and affecting a phenotypes of interest. With respect to microbiome, there are many unobserved confounders that acts together and affects phenotypes of interest. And this is something we see now in every experiment we can think of, like there are many unobserved confounders. We do not see, or like if you see these omics data set, these are, as I said, like these, all of these omics data sets only provides you only a partial view of the underlying system. So we can always think of that in any omics data analysis, this kind of, if you do it like in one omics component and the phenotypes of interest, this kind of assumptions of not observing or having many unobserved confounder can be easily satisfied, or can be something that we can, our scientists can help us say that like, okay, no, this is a valid assumption. So if you have such kind of constraint in the analysis, what what we did in this, or what we have proposed in this instrumental variable analysis approach, is basically accounting for those unobserved confounders. And obviously there are in statistics, there are already available framework or classical instrumental variable analysis framework do helps us to account for those unobserved confounders. And I'm not going in detail, but this is the classical statistical framework. What is more important in the classical statistical framework is that given that they say that we accept that there are unobserved confounders, to deal with this particular challenge, they try to look for instrumental variable which guides or which is affecting these microbiomes and not at all affecting the outcome of interest. And with that particular instrumental variable idea, now we can, so if we can generate a set of instrumental variable which only affects microbiome, but do not affect the outcome of interest, we can leverage upon those kind of framework, and that can help us to deal with the unobserved confounder issue and so on like that. So getting or motivated from the fundamental idea of instrumental variable or having instrumental variable, what this leads to experimentally. So in the larger framework of this particular study, we are looking for what is the instruments, or basically what is the, you can think of like a treatment variable. And apparently in this particular study, when she did this particular study, she basically had different introduced different genetic alterations in all of these mice. Like intervened there, the study with alcohol consumptions, like toxins, induced fibrosis and diets and all these things. Like, so these were my intervention variable. So in the larger framework, looking for accounting for these unobserved confounder, we consider these variables as our instrumental variable or intervention variable. And then we try to look for what kind of how microbiome and metabolites are associated with phenotypes in a setting like where we have unobserved confounders. Given that we were able to formulate or a classical in the larger setting of HCC already, so this particular area of instrumental variable analysis is already like more than 30 or 40, or maybe I can say 30, 40 years of old. Classically, it says that, or in the biological context, when we try to put all of these things together, it basically says that you can express a process using DAG, so directed acyclic graph, where it is saying that there are interventions or instrument which affects X, which can be our microbes or metabolites, and which in turn affect the outcome of interest Y. It can be the phenotypes of interest that we are considering. But as far as the one thing about the phenotypes of interest is that these phenotypes can be affected by some observed confounders like age and so on like that, because age, gender, and geography, basically it can affect both microbiome and metabolites as well as the outcome of interest. But at the same time, this giant H is something that is unobserved. So the framework is well equipped to handle these kind of things. The challenge that came from, or the association challenge that we are mainly looking for, mainly came from high dimensionality. Given that we are in a setting, or the classical setting is not at all tuned to handle this high dimensionality issue of this particular endogenous variable, there are settings for when the number of instruments is really high, but the classical framework is not at all good to handle when you have a lot of these things together. A lot of microbes and a lot of metabolites and so on like that. So high dimensionality is the big challenge in there, and that's where our methodology comes. And this is where so what we did, building upon our factor model or the low rank and sparse coefficient matrix idea, we took the classical framework of doing the parameter estimation. So often our interest is learning about the beta. This is where beta is something that can give us a causal or a causal association or link between microbiome and the microbiome metabolites and the phenotypes of interest, and that's what our interest is. And the good way to estimate the beta is using the two-stage least square. Given that we were mainly dealing with the high dimensionality issue, we basically replaced the first stage with the factor model, and that's where my previous work comes in, where we said we can regress X in terms of Z, but now given that we are in a high dimensional setting, we can always write down coefficient matrix as in terms of the low rank and sparse coefficient matrix, as we did it earlier, but then it improves the framework really a lot. So now I do not need to look for the direct link between X and Y, rather a combination or latent factor F, which is created as a product of X times this low rank construction, and then we can write down this V beta, which is the causal link between microbiome and metabolites in terms of the V estimated from the first stage and the gamma estimated from the second stage as a causal effect of microbiome and the outcome of interest. And something that needs to be appreciated over here is that it gives you causal effect at per microbiome and per metabolites level. So even if it is high dimensional, this framework is willing to do that. Without going and something like this is where some simulation experiment, why this directionality is super important for us. We basically in a simulated setting, we generated data corresponding to this particular tag, and then we tried to see that this is how my simulated setting looks like. And I'm interested in this particular beta thing. And in the true setting, these are the different indicators, in terms of the low rank and sparse coefficient matrix, like X times of V, Z times of U looks like. So this is the true setting generally, following the particular tag. But if we take the classical approach, where we say that, okay, I assume that I'm interested in understanding association between microbiome and the outcome of interest. And if I ignore everything else, like the instrument variable, causality and all, the available framework of lasso and things like that, where I can say that, okay, give me the microbiome data sets, it is really high dimensional. I can treat an outcome Y or the phenotypes of interest as an outcome, and in the lasso or the GLMNet framework, it can do the job, and it can give me the causal effect or the effect of X on Y by ignoring everything else. So we get one estimate. So that's like a one available framework. If we reverse the directionality, which is what the standard approach is, either using D-seq or left-seq, where you see things more from on the opposite side going from Y to X, this is what your estimate look like. So between the true and the estimated value looks like. And this is what our IV approach is like. So using our IV approach, where we take into account all the instruments, mainly the instruments, and then we see much better recovery. And to quantify all these things together, you see, if we choose the opposite directionality and use GLMNet and things like that framework, you end up with really, really large error. If you use the opposite directionality going from Y to X, then you have some error reduced, but then your error is really, really low when we actually take into account the directionality and so on like that. And that's where the improvement is. Okay, and obviously, when we applied it on this particular HCC data sets, we saw a great recovery. In the interest of time, I'll just, and the good thing about this particular result is that we saw interesting combination, if you see the simulated data result, you'll see that, Aditya, there's not much big difference between even if we have choose the directionality, but it is just that a small thing that makes a difference. So our findings when we took the experiment in the right direction, we found out interesting combination between microbiome and some of the bile acid producer and how it links together. So this is the one plot that it's trying to show. Then in the end, so I'm very much focused in multi-omics and so on, our methodology focused from multi-omics data sets, this is tree aggregated factor regression model, where we try to integrate the phylogenetic dependency that can exist in the microbiome data sets and integrate it in a multivariate framework that we have done in this particular Taro paper. We are coming up with the sequential canonical variate regression paper, where in principle, in all the previous methodology, we are considering 2 omics data at a time, and then we are trying to do regularize things or make them learn from each other, only 2 omics data set at a time. But when we are coming up with sequential canonical variate regression paper, where in principle, we can think of it, we can really leverage upon the study like TCGA and GTEx like that, where you have number of samples is really high. we believe that there are signals in there, and it will be a true multi-omics where we'll say that, okay, throw everything, all the omics component, imagine there will be some overlapping, there will be some orthogonality in there, but integrate them everything together in one framework and help us understand association or explain the phenotypes when we put all of those things together. Obviously in a sparse way, and that is something that is key. All my methodology that I presented today, it is available on my GitHub page. I'm always excited to learn about the interesting genomics data sets that gets collected. Already I'm talking to many PIs over here. They have amazing data sets, and I'm always eager to learn about it. And most of the time, my interest will be what kind of methodological or unique methodological challenge that is not there, and I want to solve it. That's my interest, and I can stop over here. I'm happy to have questions.
So you said that you are using family level, right? Like for the microbiome. You said that, but actually, like in the genus or species level, it's more like biological people.
Right.
So in the family level, even though they belong to the family level, they can function differently about microbiome. So, but you said that you chose the family level because sparsity of the data. There's like kind of gates and loss in that.
Yeah.
So, what does it mean, like why you chose the family level because of sparsity level? And the other question is that also like one of the important factor like affect the gut microbiome is actually the diet. So have you ever considered diet as a co-factor?
So to answer your first question, that's a great question. So in that specific model, we were mainly overcoming the issue of count and over dispersion. So because the data gets more and more sparser as we go at the genus and the species level, there are models like hurdle models are there, which specifically models all zero in a given generative model frameworks. There are already there. To answer your question, our focus was to model the count and over dispersion. That's why we chose it to be family. But in principle, like it can work. If you ignore the assumptions of zero or a lot of zeros, which can happen at the genus and so on like that, then basically the model works. That's what the model tends to do, or my model should focus on. To answer, what is your second question?
Okay, so it's like, have you ever considered diet?
So
Diet is the most important.
So in the model, like we did consider it. So there is so why we chose the bioscreen datasets variable, because in using the bioscreen, they were breaking it down to different, not only diet, but it's all components also. So so our interest was out of the 30,000 sample, we chose 627 just because of the notion that no, diet is super important. So that's why only focused the methodology development mainly focused only on a subset of samples.
The last time I was considering instrumental variables, I learned that it's almost impossible to find them because we need to show that they affect the X, but not the Y.
Okay, that's a great question. No, that's a great question though. So, and this is one of the main reason why when we are in the mice model setting, things are a lot in our control. I mean, like experimental wise, like we know what kind of interventions we are doing. There are lesser chance of noise. So for instrumental variable to work, DAG is super important. See, the experiment needs to follow that particular DAG going from intervention to outcome. And then obviously the bigger assumption that the instrumental variable should not interact with the outcome variables. Like that. Something, and I do not recommend the methodology for the human trial, but in the case of mouse data sets where you have control on the experiment, there with the mouse construction or the construction of instrumental variable is possible. The good thing about methodology is that if there are certain instrumental variables, so it can handle high dimensionality in terms of the instrumental variable. So if the assumption, so let's say that in a multi-omic setting, if you believe that, oh no, the intervention or the talk is happening first so when we eat something, first metabolites, then it's microbiome, and then maybe if you have, let's say, serum metabolites or serum, any phenotypes related to serum. So if you are happy to make this particular assumptions that some intervention is happening, it is going through microbiome or metabolites together, and then it is affecting our blood metabolites and so on like that. In a sense, you can make, or it is up to you that how can we make that directionality assumptions or not. Most of the time, it is in a controlled setting like mice study and so on like that. We can make that assumption, but you're right, like identifying instruments and so on like that is more constructed from the biological nature of the experiment that you have and how much assumptions that you can make. But that, like we said, there is no free lunch. So it is following the logic, like how much assumption. And so I do not recommend this particular methodology for the human experiment. But in the case of mice experiment, if things are controlled, we can use this particular kind of assumption, especially like when we know that if we reverse the directionality, we are not leading it can lead to wrong conclusions.
One last question.
Well, I was just trying to imagine how these regression models would be affected by feedback loops that come from, for example, transcription factors being themselves regulated by transcription factors, or in the case of microbiome, that the bacteria don't only interact with the environment, they also interact with each other. So is that somehow captured in the model, or is it just completely ignored?
So if it is interacting, so so we dealt with that kind of challenge through if we, statistically speaking, if you are okay saying that, okay, if two of the transcription factors are acting together, we can call it like
No, I mean feedback loops where the transcription factor is itself regulated by another transcription factor. So it's not just that they act together.
So if it is regulated by another or itself regulated by, you mean like it is regulating itself?
For example, or some other transcription factor regulates another transcription factor. This is a common situation.
So we can call it the multicollinearity problem, where if we say that like transcription factor A and B in a feedback loop are acting together, from the data centric point of view, you can say that like, okay, if we make this assumption that like A and B are acting together, we can categorize this particular problem under the larger framework of multicollinearity in a high dimensional setting, and then like so model is able to handle multicollinearity issue. But in the feedback, talking about the feedback loop, saying that like somehow A and B are acting in tandem, and if they are acting in tandem, then it falls under the multicollinearity problem, and the model can handle that.
Okay. All right. Thank you very much.